there's a revolution going on, and that revolution has collectively been called biotechnology, the genetic engineering revolution, call it whatever you will. But when you take this collection of technologies that exist now, the capacity to deliberately modify the germplasm of both plants and animals, the capacity that one has now to actually reach in and create new medicines, not primarily through the efforts of the organic chemist in the chemical laboratory, but through the efforts of the genetic engineer. When you look at this collection of technologies that we now have developing, what you have is something going on in human history that I think you could argue is certainly as important as the discovery of fire. Certainly. We have now, we now stand, I think, at the threshold of a situation where human beings have the capacity to take evolution into their own hands in ways that even so far-seeing a person as Charles Darwin never would have imagined. And so we find ourselves in a situation where the potential to do a great deal already exists, and the capacity to do even more sits on the horizon. And the question is going to arise more and more just because we can do something. The normative question comes up, of course, of whether or not, just because we can do it, should we do it? And so we have seen the growth of a whole new industry, the industry called bioethics, this industry that looks very carefully at what we can do and wonders what we should do. I want to talk about a corner of this revolution today, the stem cell corner. And of course, when I chat with my neighbors, talk to my kids sometime, and they say, well, Dad, you're going out to MIT there and hanging around the Weinberg lab where they do all this fancy stem cell stuff. This is really new, huh? And I say, well, certainly what they do is very new, but the whole business of stem cells and stem cell transplantation is one of, it's a medical technology that we've had for the past 30, 40 years. Anytime you get what we like to think of as a bone marrow transplant, what you're actually getting, of course, is a transplantation of stem cells, cells that are capable of forming every single different kind of cell that you have in the bloodstream. So stem cell technologies and stem cell transplantations, these are medical technologies that have been saving lives for a very, very long time. And what we're going to talk about today, I think, are some other stem cell technologies that have a great deal of promise. And I just as soon begin, if we can, with what we might think of as the ultimate stem cell. And the ultimate stem cell, of course, is the fertilized egg. Stem cells are cells that can differentiate into cells different from themselves. And of course, if you look at something like the fertilized egg, what you have is a single cell formed by the union of sperm and egg that can give rise to every single type of cell we have in the body. And if you're a mouse, this stem cell gives rise to all the different kinds of cells it takes to make a mouse in 21 days. If you're a human being, it's 9 months. If you're a cow, it's 9 months. Truly a remarkable occurrence. I feel a certain sympathy for my good friends and colleagues who are in the English department, and particularly those who write novels, because they have to make things up. If you're a scientist, and particularly if you're a scientist who's interested in molecular and cell biology, as we're starting now to look at the kind of things that happen in the natural world, they're so spectacular and they're so wondrous and they're so fantastic, you simply couldn't make them up. And new things come up every day. So I think the fiction writers have a very difficult time to keep up with what goes on in the biological area. So this ultimate stem cell, the zygote, gives rise to something that we think of as a blastocyst. This is an organized but small body of cells that eventually develops into all of the tissues in the animal itself. And this cell, the zygote, can not only make the animal, but it can make a placenta. So the zygote is all you need to make a creature. Now, if you look at an application of this stem cell technology that relies on the ultimate stem cells inspired by the zygote, some interesting things have happened over the past few years. You can take a cell from a normal mouse, and you can take that cell and you can combine it with an egg. But this egg has been doctored a bit. What you've done is to reach into that egg and you've taken out the nucleus. Now, a cell without a nucleus, I think as everybody knows, is very much a cell without a future. And so what you do to provide that cell with some prospects is you put in a different nucleus. But let's say this different nucleus comes from this adult cell that we've stolen from this animal. And so now, by this process of nuclear transfer, we get a cell that can go ahead in the laboratory and develop into a blastocyst. It might take it about 6 to 7 days to do that. If you hormonally treat a female mouse in such a way that she thinks she's pregnant, you can then sneak in these blastocysts into the reproductive tract, and they will go ahead and develop into a mouse. And from that kind of technology, you can actually come up with cloned mice. You ultimately get mice that are genetically identical to each other, and for that matter, genetically identical to the mouse that donated the cell in the first place. So this is reproductive cloning, where your goal is to make a creature, to make another animal, to make an animal that isn't just a phenocopy, but an animal that is a genetic copy, a genocopy of the animal that donated the cell. There's, of course, other things you can do with this technology if you don't go all the way from the stage that we've recognized here as the blastocyst stage to the creature stage. If you take the blastocyst that you create with this same kind of technology and you examine the blastocyst, you'll find there's a small population of cells, something called the inner cell mass. You can reach in and you can take that inner cell mass. Mike Kaufman, how are you? Good to see you. Yeah. You can take that inner cell mass, and these cells are what the popular press would identify as embryonic stem cells. These are the cells in the news. This is the kind of thing that all of the hollering and screaming and voting and declaiming turns out to be about. The wondrous thing about these embryonic stem cells is, first of all, you can take these cells and you can actually grow them in the laboratory. You can grow 1,000, 1,000,000. You can grow a passel of them in the laboratory. And if you grow them under appropriate circumstances, they retain their capacity to differentiate into different kinds of cells with the right sorts of inducing stimuli. People have demonstrated that you can persuade these cells to turn into muscle cells. With a different set of circumstances, you can turn them into neurons. With a different sort of circumstances, you can turn them into blood cells, and so forth and so on for many different cell types that we have in the body. So in this case, what we're talking about are embryonic stem cells from a mouse. Later on this afternoon, we'll talk about embryonic stem cells from a human. But let's stick with reproductive technologies, and let's stay with technologies that depend on the creation of a creature. And let me give a little history. About almost 10 years ago, a colleague over at the University of Massachusetts, Jim Roble, did an extraordinary series of experiments that had to do with reproductive cloning. Jim sent some of his students to the slaughterhouse where they got ovaries, brought them back to the lab, and then reached into these ovaries and brought out bovine egg cells, immature bovine egg cells that they could mature in the laboratory by appropriate sorts of incubation overnight. They could now take these mature egg cells. Remember, this kind of monkey business cloning that we're talking about depends on removing the normal nucleus that's present in the egg cell. So you can actually reach in conceptually and take out the nucleus. And here you see micrographs that actually show, and let me see if I can't do something about these lights. You can actually reach in to the egg through this tough plastic-like zona pellucida. You can stick a micropipette in, and you can suck the nucleus out of the egg. So now you've got an egg that doesn't have a nucleus. In the meantime, you have taken skin cells from another cow. You put those skin cells in culture, and they're growing very nicely. And so you take one of these skin cells up in a micropipette, and you punch through this zona pellucida again, and you deposit it right on top of the nucleus. And here you see it going on in real time here. And now, if you give a sharp burst of electrical current, what happens is you can fuse the fibroblast to the egg cell that you have, and you end up now with one cell. You actually get the nucleus of the fibroblast into the body of the egg, and you're in business, because over the course of a few days, 6 or 7 days, you get the development of a blastocyst. You can transplant that blastocyst into cattle that have been hormonally treated in such a way that they think they're pregnant, and you get fetuses, of course, and something like 9 months after you implanted these blastocysts, Jim Roble and his colleagues were able to get 3 cloned animals born. I really should have labeled this as George, Charlie, and Albert, and I have to be frank with you, I can't tell who's who. All right? Okay. But they have different personalities. George is extroverted. George will be running for office 4 years from now or something like this. George loves the camera. You just have somebody show up and George trots over toward them, whereas Charlie goes off and hides someplace, very shy. So even though these animals are genetically identical in terms of their personalities, they differ. And those of us who've known identical twins, that certainly doesn't come as too great a surprise to us. The environment, like the genotype, has a great influence on psychological development. Now, many animals have been cloned. Lots and lots and lots of different things have been cloned. And this is an incomplete list of animals that have been cloned. By far, the ugliest animal to ever be cloned was Xenopus laevis, done way back in 1952 by John Gurdon and his friends, way before the famous Dolly the sheep. Mules can now reproduce. They need the assistance of a stem cell biologist to do it, but mules now reproduce. And of course, there was this famous firm down in Texas, a cloning company called Genetic Savings and Clone that created Copycat, which is very nice. So many animals have been cloned at this stage. Now, what are you going to do with cloned animals? Certainly one of the things that you might do with animals that are used for food and for fiber is to use this technology to improve the animals. And of course, improve is a term that has to be referred to us as humans and us as our economy. From the animal standpoint, it may not be viewed as an improvement at all, but we do use these animals for food. We do use them for fiber. Whatever improvements we can make that increase them as items of nutrition, whatever we can do to make it more inexpensive to humanely raise them, these are things that make it possible for more people to eat cheaply. If you think about the kind of milk production that cattle are capable of, and I always ask my class this, and nobody knows because nobody actually works on a farm anymore. You know, how much milk does a champion cow produce? The champion milker 4 years ago, I think produced something on the order of 22 tons of milk. One cow, 22 tons of milk. And we know that that capacity is largely a genetically determined capacity. So certainly an application of cloning is to come up with animals that can produce these large amounts of milk. And people, of course, should be free, I think. If one doesn't want to have anything to do with milk coming from cloned cows, one should avoid purchasing that milk from cloned cows. But if you're someone who doesn't have quite so large an income and you would like to gain from the savings of cows that produce large amounts of milk, it might be, you might be on shaky moral ground making it illegal to sell that milk from cloned animals. But at any rate, what's going on now is that there's a lot of effort to bank semen, to select donors, and people are getting the genetic stocks together. And over the years after 2010, what we'll see are some of these animals and their products entering the food chain. And extensive analysis has been done. You can't read these tables. Don't even try to. I'll tell you what they are. They're unbelievably boring. there are the amino acid levels, numbers of certain kinds of blood cells, this kind of thing. What they've done is to take cloned animals and uncloned animals, make all these measurements, and try to see if one sees a difference. One doesn't. Just because one doesn't see a difference doesn't mean there isn't a difference there, but certainly, grossly, these cloned animals are just like normal wild-type animals. All right. So cloned animals are going to be used for food. They have been cleared by the FDA. And I want to talk just briefly, very briefly, about the use of cloned animals for therapy. We use antibodies as medicines. Everybody's heard about monoclonal antibodies because the companies that make them have made such huge amounts of money. But medicine has been using antibodies in the form of what we call gamma globulin for many, many years. Here in the United States, we use something on the order of maybe 20 tons of gamma globulin per year. We would use more if we could get it, more of it cheaply. But there's a limit to how much we can get because people are only going to donate so much blood, so that limits the supply. We also use various antibody preparations for the prophylaxis or even the treatment of a number of different infections. We would like to have much more powerful antibodies, but what we have to do is we make these antibodies, we get them from human donors. We have to use human antibodies in humans because there's genetic rejection, if you will, of animal antibodies. You can make the patient very sick indeed. And so we need human antibodies. We get these from humans. And, of course, the main thing we're concerned with is the health of the donor, not the quality of the antibody. We're not going to give additional amounts of these dangerous pathogens to these human donors in order to get a better antiserum to treat some other patient. So we're limited in what we can do by the fact that the donors of these antibodies in every case turn out to be human. Now, wouldn't it be very nice if we could actually make some of these types of antibodies in animals that had been specifically set up for the purpose? And so when Jim Roble was successful in cloning animals over at the University of Massachusetts, this is something that my wife, who's an immunologist, myself, and Jim Roble and I talked about a great deal. Wouldn't it be nice if one could use animals to help out with this antibody problem? Of course, whenever you start to talk about transgenesis and cloning to solve these kinds of medical problems, we immediately think about the mouse because we know so much about the mouse. We have so much skill in terms of handling these animals, and the mouse has served us well. But of course, if what you're going to do is use the mouse to produce these large amounts of antibody that you're going to need for prophylaxis and therapy of disease, you've got a problem. The mouse is a wonderful animal, but it's kind of small, about 30 grams. And if you're really good, you might be able to get 1 ml of the mouse's 2 milliliters of blood. And you'd need something like 150 million mice just to satisfy the demand for gamma globulin just in the United States. So even if you could raise the capital, and probably not in this capital market, but if you could raise the capital to hire enough people to bleed these mice, you wouldn't hold your labor force for any more than a week. It would be terrible work. On the other hand, if you look at an animal like the cow, this beast can weigh a ton. It has 60 liters of blood. You can collect something on the order of 10 liters of blood every month from a cow, and it doesn't get anemic. So with about 5,000 appropriately genetically engineered cattle, you could very likely satisfy this demand. So what we did back in 1998 was with Jim Barton, a lawyer friend of ours from Connecticut, we formed a company called Hematech. And the purpose of this company was to engineer cattle to make a variety of different human immunoglobulins. And just in an overall sense, not to bore you with too many of the details here, what was done was to take the collection of genes that encode human antibodies and put this collection into a cow cell, and then take enucleated bovine eggs and force them to use the genetic information in this genetically altered cow cell to produce cattle. And when we tested the serum from these animals, we found that they did indeed make human immunoglobulin. And in the years since this work was done, our partners, our Japanese investors, have been able to completely abrogate the cow's capacity to make cow antibodies, and the cows only make human antibodies. And so the expectation is that at some day in the not too distant future, you'll see these showing up in medical applications. Of course, we sold the company, I guess, 3 years ago, and the checks they send don't bounce, but we don't get much information, so I can't tell you exactly where the project is now. But I only offer this as an example of what can be done in terms of genetically altering the stem cells of animals, if you will, and then using those genetically altered stem cells to make cloned animals to serve a useful purpose. Let's go back to the subject of therapeutic cloning. And now, instead of talking about mice, let's talk about human beings. So the same kind of experiment we talked about with mice, a situation where you take a cell from a human being, a skin cell or fibroblastic cell, and you introduce the genetic information in that cell into an enucleated egg, allows you to form a blastocyst. And now you can go into that blastocyst and you can harvest from that blastocyst cells that will sponsor the development of embryonic stem cell cultures. And these embryonic stem cell cultures are capable of doing just what the ones were we talked about before. You can make cells of the blood. You can make nerve cells. You can make muscle cells. In fact, cardiomyocytes, heart cells, have been produced from these embryonic stem cells. And you can make pancreatic islet cells from them. So these cells are capable of performing a variety of different functions that are of very clear medical importance. The problem, of course, is that when you take this fertilized egg and you use it to develop a blastocyst, in order to get the embryonic stem cells, you have to destroy the blastocyst. And it's quite possible that under appropriate circumstances, that if you had nurtured that blastocyst within the womb of an appropriately prepared mother, that this blastocyst could 9 months later give rise to a human being. So you have a conversation that's different from the conversation you had when you do this kind of experiment with a mouse or with a cow or with a sheep. Because now what you have is the potential of human life as represented by this blastocyst. And whatever one's feelings about properly honoring this potential for human life, there are some that say it should not be destroyed. There are others who say that at this stage, and if the prospects are such that it would never enjoy a human life anyway, that for purposes of therapy, it would be perfectly all right to use them for this purpose. Whichever side of that conversation, that argument, that ethical debate you come down on, we have to realize that it's there and that it's different from working with mice. As we move back from that particular debate here in the United States, what we also have to realize is that different societies, different cultures will view that debate differently. We may feel one way about it here in the United States. We may be split in our feelings here in the United States, but there are other societies in the world where a similar split has not taken place to that degree, and people are proceeding apace with this kind of research. What we have to face up to here in the United States is whether we decide to do this as intellectually aggressively as we can, there will be other societies, other cultures, powerful societies and powerful cultures that we respect who will go forward, who are going forward full speed. But that's a conversation that belongs in the philosophy department. We're talking about biology here, so let's go on. There is another way that people have come up with, and this is kind of an extraordinary story. When you took chemistry, remember you heard about the alchemists, and the alchemists wanted to take base metals and they wanted to turn these base metals into gold. Silver would do, but preferably into gold. And they worked in all kinds of creative ways to do the impossible. There's a kind of alchemy that people in developmental biology have for many years wanted to do. What a developmental biologist would have loved to have done, would have dreamed of done, would have fantasizing about doing just a few years ago, would be to take a skin cell or a muscle cell. Let's settle with just a skin cell. It would be to take a skin cell and turn that skin cell into the kind of stem cell that could form any type of cell present in the body. In other words, turn that skin cell into something very much like an embryonic stem cell. Because if you could take a skin cell from me and turn it into an embryonic stem cell, then there's a possibility that you could develop pancreatic islet cells that I might be able to use, that my immune system would accept as self and hence wouldn't reject. There's a possibility that you could develop cardiomyocytes that I could use to repair areas that had undergone ischemic injury during a myocardial infarction, during a heart attack. And my immune system wouldn't reject those cells. And I think you get the picture. So it isn't just that it would be a neat scientific trick, that it would be an extraordinary scientific feat. Here we're talking about something that is, from a medical standpoint, extraordinarily practical. So I tell my students, that if you're going to be a successful scientist, you've got to be very social, because you go to meetings, and at scientific meetings you hear lots of talks where people turn the lights off in the room and put lots of stuff up on boards and yak at you a lot. But after we do that, we tend to go to the bar, we tend to go to the dining room, and we have a lot of wine and beer. Not a lot of hard liquor. Scientists are funny. We're not great Scotch drinkers or gin, but we drink a lot of beer, and we love wine. And so you sit into the night and you go over ideas, what so-and-so said, what you'd like to do, and that kind of thing. Everybody talked about how they would like to turn skin cells into embryonic stem cells. But everybody knew that that would be an extraordinarily complicated thing to do. It would probably have to be done in several stages, and that maybe sometime during the next 20, 25, 30 years, maybe people would be able to approach that problem closely, and maybe somebody might be able to at least do something a little bit like that. The laboratory of Yamanaka in Kyoto, Japan, at Kyoto University, dropped a bombshell just about 2 years ago. Two people, Takahashi and Yamanaka, took just 4 different genes. Just 4. People thought there would be scores of genes that would be necessary. Took just 4 genes, introduced them into fibroblasts, the equivalent of skin cells, as I'm chatting about them here. And they were able to, they did it first with mice, and they were able, and then later with humans, and they were able to induce these skin cells to become pluripotent stem cells. And these pluripotent stem cells that people have now been studying very, very actively all over the world now, they will do very much what the embryonic stem cells that I showed you before will do. They will turn into many of the different cell types we find in the muscle. People have turned them into cardiomyocytes, heart cells, heart muscle cells, if you will. They've turned them into neuronal tissue. They've turned them into hematopoietic cells. They are, for all practical purposes, embryonic stem cells, or cells that behave very much like embryonic stem cells. They neatly avoid the moral argument that we made before about the necessity of destroying a blastocyst that had the capacity to develop into a human being. So it was a way around that moral barrier that many people felt were terribly important. So there are two ways of doing what's thought of now as therapeutic cloning. Of course, there are many who feel, and I would say they feel this way appropriately, there are many who feel that this area is very much at the beginning. It's at the beginning of the beginning. No one knows which type of cell, the type induced very artificially here in the Petri dish totally, whether this method produces the best hematopoietic cells for reconstituting the blood system or the best cardiomyocytes, or whether this system produces the best cells for making cardiomyocytes, but the other system produces the best one for nerve cells. The only way to know will be to conduct the experiments aggressively and in an unfettered manner. If what we do is to decide that what we're going to do is we're going to do it one way and not do it another way, we are effectively tying the hands of researchers. But we're selectively tying the hands of researchers. We're tying the hands of researchers here in the United States. They're unbound in Singapore, absolutely unbound. All right? So the work will go forward. The question is, where will it go forward? Now, and I'm not going to go on and on, but I have to tell you a couple more things. There's an extraordinary demonstration of the power of this therapeutic model that's been done by Jaenisch and his colleagues at the Whitehead Institute at MIT. There's a terrible disease known as sickle cell anemia. Sickle cell anemia is a disease that, in particular, affects people who trace an ancestry back to the west coast of Africa. In the American black population, you have an incidence of sickle cell anemia of about 1 in every 350 to 400 births. So it is a severe health problem that afflicts the black population very strongly here in the United States and even more strongly on the west coast of Africa. The sickle cell gene is a gene that evolved, as many of you know, as a protection against the malarial parasite. And it did work as a protection against the malarial parasite. But DDT and quinine are much, much better and don't have the same sequelae for the population that you get. As long as you have just one copy of the sickling gene and a normal gene, you're fine. In fact, you wouldn't know you had a copy of the sickling gene unless you had a very sophisticated analysis done for it. It's when you get those two genes together in one individual and the person is homozygous for them, both copies, the one you got from mom, one you got from dad, or sickling genes, that you then develop sickle cell anemia, a very, very severe illness. At the Whitehead Institute at MIT and other places, they work with a mouse model of sickle cell anemia. These are mice that have been genetically modified in such a way that they get sickle cell anemia, if you will. All right? Okay. What was done by the Jaenisch laboratory is to take fibroblasts from these mice, and then in the laboratory, they turn these fibroblasts into the equivalent of embryonic stem cells. These induced pluripotent stem cells, the equivalent of embryonic stem cells. And then in the laboratory, they genetically modified these cells so that they, in effect, removed the sickling gene and put in a normal gene for hemoglobin. They then redifferentiated these cells into blood-forming cells, and they injected these cells into the mice and cured the disease. So in the mouse, what you have is an example of therapeutic cloning with the onlogin, if you will, of the capacity to turn cells into embryonic stem cells well laid on. And the sort of thing you get is a model for what we all want to see done with human beings. One more thing. This is a photograph of a liver taken from a patient who had cancer. And it's metastatic cancer. This is not a picture of the primary tumor. This is a picture of the liver to which lung tumor cells have metastasized. Now, just a very few years ago, if you had asked anyone doing cancer research, which one of the cells in this particular metastatic lesion or this particular metastatic lesion give rise to cancer? Here they are looked at much higher magnification. Which one of these cells can give rise to cancer? The answer would have been any one of them. Any of them could give rise to cancers. Made sense. Well, but there had been troubling observations that had been kicked under the rug for years and years. Here's what you found in the laboratory. In the laboratory, if you took tumor cells and you injected an experimental animal with 10 or 20 tumor cells, you didn't get a tumor. If you injected 100, you didn't get a tumor. 1,000, you didn't get a tumor. 10,000, you rarely got one. You only regularly got tumors when you injected large numbers of cells. The usual number was something on the order of 1 million. Right? Well, that's because you didn't inject in the right spot. It's because you push the cells through the needle, a number of them get damaged, because you didn't handle the cells correctly. There are 1,000 reasons why people swept that observation under the rug. Never was accepted until just a very few years ago that maybe all cancer cells aren't equal. Right? In 2003, an extraordinary paper was published in Proceedings of the National Academy of Sciences. Here's what people thought, that if you take any cell out of a tumor, any one of those cells can cause cancer. What this group did, what Mike Clark's group did at the University of Michigan, was to take cancer cells and take advantage of the fact that on the surface of the cells, all cells, you have a profile of many, many, many different protein molecules. And it turns out that for different cell types, for different kinds of cells, there are specific profiles of these proteins that are as useful for identification as a set of fingerprints might be. And you can use these protein profiles to identify one type of cell as opposed to a different set of cells. You can even go farther, because if you go within a broad category of cells, what you find is that these cell surface profiles allow you to even recognize subpopulations within that group of cells. And so what they did with the population of breast cancer cells was to look at the cell surface profile and notice that some of these cells had differing profiles. To make a long story short, they noticed there were populations of cells that had rather high levels of a protein known as CD44 and rather low levels of a protein known as CD24. Whereas most of the cells were just kind of all over the map. Most of the cells were high for CD44 and high for CD24. So what they did was to separate this breast cancer cell population into those that were CD44 high and CD44 low and all the rest. And they made the very dramatic discovery that if you took this population of cells and put them into a mouse didn't get cancer. You could put a lot of them in, 20,000 of them in. If you put just 200 of these cells into the mouse got cancer every time. Didn't take many cells at all. That was one of the several observations that have led to the notion that cancer has stem cells. There are cancer stem cells. That cancer as a whole is made up of many different cell types. But these cell types can divide a few times so that you get a bigger tumor mass, so you expand the mass of the tumor. But if you took one of these and let it go into the bloodstream and move to a different part of the body, that one couldn't initiate a new tumor. It couldn't cause a metastasis, if you will. Rather, you have to have cancer stem cells in order to actually propagate the tumor to a new location. If you kill off all of these cells, but you leave the cancer stem cell population, the patient will certainly get better. You'll shrink the tumor mass enormously, but the tumor is very likely to come back because you've left the stem cell population there. So people have now looked for cancer stem cells not just in breast cancer and not just in hematopoietic cancers, but they've looked for them and found them in breast cancer, colon cancer. Actually, they were first found in leukemias, prostate cancer, a number of different sorts of brain tumors, pancreatic cancer, melanoma, and the list gets longer every 2 months. So now we have the notion. We're stuck with the information that all cancer cells aren't equal and that tumor therapies are probably going to have to be designed so that they not only shrink that mass of cells that represent the majority of the cells in the cancer, but we're also going to have to be sure that we're developing therapies that will attack the tumor-initiating cell population, the cancer stem cell population that's present in the tumor. Because if we don't do that, according to John Dick, one of the pioneers in this area, it would be very much like trying to clear dandelions out of your backyard. If you just cut the leaves off, but you don't kill the root, you're going to come back a couple of weeks later and you're still going to have a backyard full of dandelions. Right? So I think I want to leave you with the following message. With regard to stem cells, the bottom line is stay tuned. There are going to be lots of developments. Okay, I think I've yacked at you long enough. So, your comments, questions, please.
Hey, can you give us a hint of what kind of stimulus it takes to produce particular kinds of cells from a poor potential cell? How you make only muscle cells or only blood cells from it?
Yes. Recently, there was a very dramatic article that came out in Nature from the Melton Laboratory. And what the Melton Laboratory was interested in doing, they were interested actually in taking one type of cell present in the pancreas and turning it into an insulin-producing cell, a beta islet cell. And for years, people have been looking at what kinds of genes are expressed in beta islet cells as opposed to the genes expressed in non-beta islet cells. And they came up with some candidates. And so they took one of these candidate genes, transfected it in, and were able to effect that transformation. If you're trying to make blood cells, cells of the hematopoietic system, people have studied for a very, very long time what kinds of intercellular hormones, we biologists would call them cytokines, what kinds of cytokines are necessary to do that. You can steer, to some extent, the development of these cells by putting the right mixtures of cytokines in to get particular types of blood cells. So this is a business where it's very much like, it's a sort of, if you will, developmental agriculture. You do need to know what kinds of ingredients, what kind of influences to give in order to do this kind of steering. And people have made a great deal of progress with that. Yeah. Mm-hmm. Yeah. Please.
We have a son who's been diagnosed as a 17-year-old childhood diabetic. And we have been told that stem cell research was designed by some doctors at Northwestern, and it's been brought to be executed in Brazil. And that work is being done there. Are you aware of those studies?
Could you repeat the observation for me, please?
Yes. There's stem cell research that was designed here in the United States, could not be executed here, working with childhood diabetes, that is being executed in Brazil. And they're working there on the cure for childhood diabetes. And I was wondering if you were aware of that research.
I am not aware of that research. And I'm going to take from your comments that this is research that could not be executed here in the United States for ethical reasons. I see. Yeah. I'm sorry, I'm not aware of that work. Not only Brazil, but in some other areas of the world, they're doing a good deal of work. Every place, fortunately, won't allow you to do just anything. All right? There are constraints in other parts of the world too, but in some places they are looser. Here's what we do know, that if someplace else, if in Singapore, if in Britain, if even in Brazil, someone comes up with a technology that can be demonstrated to actually work, first of all, we'll hear about it widely. The people who do it will be very much lionized because they've done it. And that will provide additional arguments for us to change our policies here in the United States. In this field, information doesn't stay compartmentalized. Please.
I was just going to add, Professor Milton's lab at Harvard does not operate under these constraints because it's funded by Harvard. And Milton is working on exactly the childhood diabetes issue he was just describing from that. And if you had questions, for someone in the U.S. about the Brazilian research, I would turn to Harvard stem cell program because they're working in that area.
The Harvard Stem Cell Institute is one of the premier ones in the world, and what's been pointed out here is that here in the United States, if you use money that you get for research from a grant from the National Institutes of Health, a governmental agency, there are a set of constraints that you have on your work. If you have funded this work privately, and a good deal of the work done at Harvard and some other places is privately funded, then you're not under those same constraints that the federal government imposes. Fortunately, Harvard and Stanford and MIT and our other great research institutions still don't allow people to do just anything they want to because it's not government funded. Fortunately, these institutions have human review boards that will examine any kind of experiment that's going to use human subjects. And with stem cell research, everybody is very, very sensitive to the ethical implications. And so even in places that are funded privately, places like Stanford or Harvard, fortunately, procedures still have to undergo levels of ethical review. And I think that you wouldn't want it any other way. Yeah. Other comments or questions here? Please.
I guess I'm not totally understanding in terms of stem cells. Isn't there also a concern that if you were to treat someone that has childhood diabetes or else with stem cells, that their own antibodies in their body would try to go after that, and so you have to immunosuppress those people?
Well, the great promise of stem cells, of the kind of therapeutic cloning we talked about here, the great promise that you have is that you could use cells that are from the patient, and you could turn those cells into the appropriate cell type you needed for repair. So the immune system would recognize those not as foreign cells, but as part of self.
As long as they go through the embryo. Then we're talking about embryonic, or we're talking about
now there are two ways to do it, right? One way to do it is to actually take a cell from this individual, put it into an enucleated egg, develop the blastocyst, and get the inner cell mass from which you could develop a stem cell culture, right? That would be one way. Another way would be to take a skin cell from this person, to add the appropriate mixture of genes, and now convert it into one of these induced pluripotent stem cells, all right? In either case, you would create cells that would be self cells and so would be accepted by the immune system. Here's what I didn't tell you. I didn't tell you that as wonderful as this new technology is for taking skin cells and turning them into something very much like embryonic stem cells, what you do is the routine procedure is to take 4 genes. A gene called MYC, a gene called SOX2, a gene called KLF4, and a gene called OCT4. MYC is a very famous gene. It's a very famous gene because it's a gene that cancer researchers have studied a great deal, because if you put forms of MYC into cells, you can turn them into cancer cells. And Kreupel-like factor 4 can also act as an oncogene. And so this procedure involves the addition of genes that can predispose the cells to a malignant transformation. And so the procedure is great for showing in principle that this can be done, but nobody wants to practice medicine today where they would be changing cells using these particular genes. There's another trick they have been using too that you wouldn't want to do, and that is the way in which you get these genes into the cell and get them expressing is you attach them to something called a retrovirus. All right? And there are a number of tumor viruses that are retroviruses. HIV happens to belong to the great family of retroviruses. And so whereas these are wonderful experimental tools for introducing genes into cells and studying what effects those genes might have, this is definitely not a technology ready for the clinic at the present time. The good news is people are forging ahead. This is a problem that everybody recognizes. People are working very hard to get alternative methods of introducing these genes. People have already found that you can get some of these transformations without putting in the MYC gene that I had mentioned a little earlier. There's some other groups that are able to dispense even with a couple of the other genes there. So I think we can look forward to, if we stay tuned, we can look forward to great methodological improvements that will make this induced pluripotent stem cell manufacture a much more safer route to take than it is right now. Now we're looking at a new concept that's been realized in the laboratory. What we really want to see over the next few years is a technology that's safe to use in human beings.
Yeah, I think this is absolutely marvelous. I think that dangerous, the popular press makes it sound like it's going to happen tomorrow. So, your cautions should be there too.
Yeah. Yes. Please.
I know you've talked a lot about the positive aspects of this research. But don't you think there is a huge open window for abuse of this technology? And can you just elaborate on that and what could possibly happen? Because I feel like there's so much. While it is a great and marvelous technology, there definitely is that slippery slope argument.
The ethical arguments against therapeutic cloning are many, from the basic argument that once you have something that could be developed into a human being, that gives it special status and special interests, and that it becomes inviolate and sacrosanct. That would even be true for embryos that have been created for fertilization in vitro fertilization clinics. As a rule, if you're going to undergo in vitro fertilization, what happens is the mother is sort of superovulated and then eggs are withdrawn. That's a procedure that has a number of medical risks associated with it. It's done under anesthesia. And so if you're going to go in and harvest eggs, you don't want to do that 2 or 3 times. You want to do it only once. And so you take a lot of eggs and you fertilize a lot of eggs. Every egg you fertilize doesn't actually develop. And so there are a few there in the Petri dish that have developed quite well. And so what you routinely do is you implant maybe 2, maybe 3, so that you don't have to put the mother through this procedure again. And in case those don't work, you save the rest. You save them in frozen form. And you might save 10, 20 such embryos. You know that you are not going to use all of those embryos in this mother. You know that. There's a good chance you'll never have to use those embryos again for this couple that you're helping. But you do this to avoid the additional risk to the mother. There's only so much time that you're going to spend the money to put the liquid nitrogen into the tank that will keep these embryos frozen but viable. Eventually, you're going to have to discard these embryos to make room for more. There are responsible arguments that would refer to that as killing, and there are responsible arguments that say it's no such thing. One way to organize society is to have the diktat of people who say it's no such thing, have that imposed on the entire society. Another way is to have the diktat of people who say it is killing imposed on the whole society. We as a society are working our way through that normative argument now. All right? Please.
The current types of cancers have a mixture of the tumor cells and the stem cells. Now, our current chemotherapy protocols are not as effective against the stem cells as they are regular cells, and therefore maybe we never kill the stem cells in some of these tumors. We have to get newer techniques.
People are now starting to research that topic very aggressively, And a couple of things that people are finding out. One is that some stem cell populations appear to be a little more resistant to radiation than other populations are. And many of the chemotherapeutic agents that we use are agents that kill cells, that cause cells to undergo cell death. And one of the things that people find is that a major mechanism of doing that is to cause damage to the DNA of the cell. The cell recognizes that damage and then commits suicide, all right, undergoes a process known as apoptosis. Stem cells typically don't divide as often as other cells do, and so they are not as susceptible to many of these agents, such as radiation or other cytotoxic agents. And so that's exactly what one finds, is that some of the conventional agents and treatments that are used, stem cells are probably not as susceptible to those treatments as others. So what people are doing now, that one knows about the existence, one has assays for these stem cells, what one does now is to set up programs deliberately to try to develop and try to identify elements that will affect these stem cell populations. Right. Absolutely, absolutely. Yeah. I'm fortunate enough to spend time at the Whitehead Institute from time to time in the laboratory of Robert Weinberg, who's one of the great figures in this field. And Robert Weinberg, whose mother died of multiple myeloma and who's one of the world's most famous cancer scientists, says there are probably more people making a living off cancer than there are dying from it, because it's the kind of field that always generates new questions, new things to do, new approaches, and there just isn't a research institute research enterprise in the world that is supported at the level of cancer research. That would go for high energy physics, would go for any area of biology you want to take. Enormous amounts of money are being spent, enormous amounts of research are being done, but as you probably know, the questions are really tough ones and new ones keep coming up. Yeah. Please.
Are you familiar with the work being done by Cole Isaacson at Lane Hospital?
What work is this, please?
Embryonic stem cell research. They're producing dopamine-producing nerve cells.
Yes. There are a number of groups that are very interested in nerve cells that can produce dopamine, because these are the kind of nerve cells you want to use for Parkinson's disease. So it's a very, very active field of work. Yeah, absolutely. One more question, and then I'll let people go. Absolutely. Yes. I've got the signal. Shut up from my boss here.
I'm just curious, assuming you've been able to generate most of these therapeutic cells,
yes,
what would the methodology be? How would you safeguard? In other words, how do you know what the correct dose would be of these self-generated stem cells if you were trying to repair heart damage or whatever? How do you then, what's your methodology?
Suppose someone's able to generate stem cell populations that are perfectly safe from a safety standpoint. How would one now go about getting these into the clinic? It would be like with any other experimental treatment. You would have to set up clinical trials. You would use graded doses of cells. First thing you would establish is how many cells can you give without doing damage. And once you've gone through that phase one, then you would set up situations to find out if giving these cells actually helped the condition. So you'd need control and experimental groups, and it would proceed through the normal course of clinical trials. It would take a long time and would cost a lot of money. Yes.